Hey guys, welcome to what's turned out to be the 6th video out of 6. I thought it was going to be a 5-video series, but then I decided to split out second messengers because they're a little tricky, a little complicated, need a little more time to explain. But here we are in the final stage, stage 3 of cell communication. Our receptor's been activated, the signal has been transduced, and now we need some cellular response proteins to get involved. The cell needs to respond somehow, and hopefully I've hinted at some of the different cellular responses along the way, but let's take a little closer look at how the cell might respond to its signals. So receptor's activated, and we got some kind of signal transduction pathway, and then a cellular response. And there are 3 main proteins that are the targets of these signaling pathways. There are enzymes that can be switched on or off. There are structural proteins, for example, excuse me, wow. There are structural proteins like cytoskeletal proteins that can get the cell to actually change its shape, whether it's a muscle contraction or something else. And there are what we call gene regulatory proteins. Another name for these are transcription factors. You've heard me hint at these in a couple of the examples I've given so far, where the cellular response involves changes in gene expression. And it's these proteins that are either activated or inactivated in order to alter gene expression. So let's take a look at each of these targets briefly so we understand how a cell might respond to a signal it receives. We've talked a little about kinases and phosphatases. We've talked about how a protein kinase can take ATP and hydrolyze the third phosphate, which gives it both the phosphorus it needs to attach to its target protein, and also the energy it needs to create those new bonds, right? You need both energy to create a bond, and you need the raw material. And so by hydrolyzing the third phosphate off an ATP, you get both the energy and the phosphate for it. In most cases, most of these protein kinases are what are called serine, threonine, or tyrosine kinases, meaning that they're targeting one of those 3 amino acids in the target protein specifically. And then protein phosphatases, we know, are plucking that phosphate back off again in order to make sure that the cell response and the signal transduction aren't somehow permanent within the cell. That would be bad news, right? Transcription factors, these gene regulatory proteins, are really important targets of signaling pathways. So, for example, transcription factors that are called activator transcription factors will bind to a sequence of DNA just upstream of the promoter called an enhancer. And by binding to the enhancer, they actually enhance the binding of RNA polymerase at the promoter, and therefore enhance the transcription. They upregulate the expression of that particular gene. A repressor transcription factor has the opposite effect. It binds to a sequence upstream of the promoter called a silencer. And when that repressor transcription factor is bound to the silencer, it blocks RNA polymerase's ability to bind to the promoter, and therefore it suppresses the expression of that particular gene. Some genes will have both enhancer and silencer regions, sometimes called repressor regions, upstream of the promoter. Sometimes they'll just have one or the other, depending on how that particular gene's expression is regulated. Check out on YouTube, look for DNA partitioning during mitosis, and look at all the complex steps involved, and think about how signals might be regulating the different responses. Same with find a video of an amoeba crawling. Go towards nutrients or away from something negative. Or white blood cells called macrophages chowing down on bacteria. In the case of the amoeba and the macrophages, the cells have to change their shape in order to do that. And that means changing structural proteins very rapidly. DNA partitioning during mitosis requires a complete rearrangement of the cytoskeleton. All of these are examples of cell signaling leading to structural protein changes. So we've said before, earlier in the class, that cytoskeletal filaments are in a dynamic state. They're not static. Actin filaments can add new actin subunits at one end. They can shrink at another end. They can grow and shrink again that way. Microtubules can do the same thing, where we can be adding microtubules in the form of tubulin dimers, or we can be depolymerizing them as well. And it's true with the intermediate filaments, that they can be polymerized together, extended, shortened, all in very rapid succession. And don't believe me? Watch an amoeba crawling in real time and recognize that that is very rapid changes. What you're seeing is very rapid changes in the cytoskeleton itself. And so in this case, you've got cytoskeletal filaments either growing or shrinking due to, typically, phosphorylation from some sort of a cascade that's taken place. So the cell response proteins. So our cells are going to respond either, most commonly, either enzymes are going to be switched on or off to active or inactive states. Gene regulatory proteins, transcription factors, either repressors or enhancers or activators, right? Or cytoskeletal filament proteins being extended in one direction, shrunk down in another, etc., in order to cause movement or physical changes to the overall cell. Okay, I hope this series of videos has been really helpful to you as you've thought about how cells communicate with each other, and then in particular how they receive and respond to the communication signals that they get. Thanks, guys.